Hello friends, welcome to our YouTube channel, and today we will be discussing about another topic that is called as the formation of the anchor cell. Till now we have studied regarding the formation of vulva in the C. elegans, and this formation of the vulva was mediated with the help of the cell that is called as the anchor cell. Sometimes the questions is also asked in CSIR net examination regarding this formation of the anchor cells. So let us take a look on the topic. Okay. So in the gonad region there was an anchor cell. In the gonadal region there was an anchor cell. Okay, and below this there were 6 vulva precursor cells. 1 2 3 4 5 1 2 3 4. There were 6 vulva precursor cells, and those were 3 P, 3P, P4P, P5P, P6P, P7P, and P8P. So there was 6 cells, and above all these cells there was this is a gonad, this is gonad, and gonad is somatic. And there was a cell, and that is called as the anchor cell. There was a cell that is called as the anchor cell. So today we will be going to discuss about what was the genesis, what were the factors responsible for the development of the anchor cell, and at what stage of the development this anchor cells was formed. Okay, so we will be discussing here regarding the same. So let us understand during the course of development, in the larval state of the C. elegans, what actually there was the 4 cell, that is from Z1 to Z4. Okay, from Z1 to Z4. Okay, so it was named Z1 to Z4. So Z1, Z2, Z3 and Z4. Z1 and Z4 was acquired somatic cell fate, and it was a descendant of MS. Okay, and after series of the development, after the series of cell division, okay, it acquired this structure that is called as Z1.ppp and Z4.aaaa fate. P stands for posterior, A stands for anterior. Okay, whenever suppose the cell divided, and that is called as, suppose after division EMS was formed. Then after EMS was formed, it again divided to E and MS. Okay, and then MS went through subsequent division. MSP means posterior, MSA means anterior, and in the subsequent division the genesis of the Z1.ppp and Z4.aaaa occur. Then what about this cell, that is Z2 and Z3 acquired the germinal fate. It acquired germinal fate, and it was a resultant of P4 division. P4 act as a somatic cell, so it was resultant of the P4 division. Okay, so we will try to proceed with these 2 cells. Okay, and that is called as Z1.ppp and Z4.aaaa. So out of 4 cells we have the 2 cells, that is called as Z1.ppp and Z4.aaaa. Okay, so we have these 2 cells. and these two cell will undergo the development of anchor cell. Okay, this two cell will undergo development cell, but which one, either this cell or this cell will go for the development of the anchor cell. Okay, because there are two cells, then only one cell will undergo the development of the anchor cell and another cell will go for the ventral uterine cavity or ventral uterine body. So this is a big question here, and here on the basis of some sort of the signaling or some sort of the protein and its receptor formed, okay, and this will determine that which one cell will undergo the anchor fate and which one will undergo the formation of the ventral uterine body. Okay, so let us take so let us see here. There initially, at the initial phase, if we talk about the initial phase of the development of the anchor cell, okay, these two cells, that is Z1 okay, these two cells, okay, that is Z1.ppp or Z4.aaaa. Okay, so let us understand here, these two cells will form the two different protein, and that is called as the lean 12 and lag 2. Okay, lean 12 and lag 2. The two protein will be formed with the help of these two cells. Okay, just two cells, and which cell will become the anchor cell, it is right now no clear. It is totally a by chance process. Okay, so I haven't labeled any cell with its own name, either Z1.ppp or Z4.aaaa. So it's a reshuffling process, it's a random process. Okay, so this cell is also secreting the lean 12 protein and this cell is also secreting the lean 12 protein. Remember, the lean 12 protein was also responsible for the lateral inhibition of the cell, fate from which it was secreted. If it was secreted from the 1 degree of the vulva precursor cells, then it will stop the 2 degree of the cell to become the 1 degree of the cell. So it was also involved in the lateral inhibition of the cell. Okay, so right now here there is another the same lin 12 protein is there and another same, but what actually happens here, these two proteins are secreted in very minute quantity. These two proteins are secreted, which one? And that is called as lin 12 and lag 2. These two proteins are secreted in very trace amount, but after subsequent time or division, what actually happens? Okay, so this is an first step, you can say this is a first step, and it is also called as the initial step of anchor cell development. Okay, then what actually happens then after? The second step is stochastic effect, means the random event or by chance event. What happened here, at certain step after the development, what happens, the formation of lag2 protein and lin2 protein, the lag2 protein and lin2 protein will increase, and this increase in the formation of this lag2 protein and lin12 protein will have some fate. And what is that fate? Let us understand. Okay, and before going through, let us understand in the coming time what will happen. The lag2 protein will be a protein, and the lin12 protein, which is being secreted by both of the cells right now here, lin12 protein will act as receptor protein for lag12. Okay, so right now here what happens means the lag2 protein, lin2 protein will act as a receptor for the lag, and again here okay. Now I have made a diagram, arrow diagram, means you can say that lin12 protein is acting as, lin12 protein will act as a receptor for the lag2, and here you can say means the here lin12 protein will act as a receptor for the lag2. Okay, but stochastically what will happen here means this cell will start to make, these two cells start to make excess amount of or abundant quantity of lin12 and lag2 lin12 and lag2. So okay, so what will happen here, suppose this is a lin12 and this is lag2, lin12 and lag2. Okay, then what will happen actually here, let us understand here. Okay, now what will happen, one of the cell will start to produce lag2 in more quantity, and another one of the cell will start to produce the lin12 quantity in excess amount. Okay, and this is a stochastic event, which cell will produce more, it is unpredictable, it is completely unpredictable. So I have seen, I have just shown in a diagram that suppose this is a lag2, means this is the lag2. So I have made the 3 arrow, means I have made the 3 protein which will encode lag2 and lag2. Okay, and I have made here the receptor area, lin12 and lin12. With this figure, okay, you can easily understand that this cell is producing more amount of lag12, and this cell is producing more amount of lin12 protein. Okay, so those cell which will express more amount of LAG2 will be the future anchor cell, and those cells which will produce more amount of LAG12 will be responsible for the ventral uterine cavity. Okay, first. Second, lean 12 and the lag2 just acts in opposite direction, means those cell which will have excess amount of lag2 protein will inhibit the formation of the lean 12 protein, or vice versa. You can say those cell which is having lean 12 protein will inhibit the formation of or the transcription of the lag2 protein. So this is what the things appear here in the second step, that is called as a stochastic effect. Okay, now, and this one here, this one here, you can see from the diagram that lag2 is having the lag2 is having the 3 proteins, and it is being received by the lean 12 protein, and only one lean 12 protein is there which is being accepted by the lag2 protein. Okay, so it means that the lag2 protein is what? Lag2 protein is giver, it will give the protein, and the lean 12 protein is the receiver. Okay, so what could be the conclusion? Lag2 protein is giver, will give, and lean 12 protein will act as receiver. Okay, and lag2 protein okay will inhibit lean 12 expression, and lean 12 will inhibit the lag2 expression. In this way, lean 12 will stop the expression of lag2 protein. So it is very clear in the figure what we have expressed here. So in the next diagram we can see that this is called as the feedback inhibitory step, and the third step is feedback inhibitory step. Okay, and in this step we will find that this is nucleus. We will find that so here was the expression of lag2 protein, so lean 2 protein. Okay, this expression is reduced, and here the lag2 protein is expressed more, or you can say that it is increased, and this one is decreased. Okay, and in this opposite cell you will see that the expression of lean 2 protein is increased, lean 12 protein is increased, and lag2 protein will be decreased. It is very clear right now. Then what will happen here? Again, this is a lag2 protein, this is a lag2 protein, and it will produce more, and this is a receptor. This is a receptor, that is the lin 12. Okay, this is a lin 12 receptor, and again this is a lin 12 receptor. Again this is a lin 12 acceptor. Again this is a lin 12 acceptor. Again this is a lin 12 acceptor. Means you can say here in this figure that the lag 2 is being produced by this one cell and the lin 12 is produced by this two cell, means A cell and B cell. Okay. Now it is clear, and it is also clear that the lag 2 will inhibit the formation of the lin 12 protein in this cell, and the lin 12 protein will stop the formation of, will inhibit the formation of the lag 2 in this B cell. Okay, means they are you can say that they act antagonistically. Okay, so if you want to say they will act antagonistically, then you can describe it here as this lag 2 protein, will act as antagonistic to each other. Okay, will stop, and those cell which is producing more lag 2, then it is called as the pre-VC fate, and those cell which is having more, which act as a receiver. Okay, this is, you can say, giver. Okay, giver, and this one cell you will be called as the pre-VC, pre-uterine VU, ventral uterine. Okay, and this is a receiver. Okay, and then after subsequent division, after further division, okay, it will acquire the anchor cell. You can say this is not VC, this is anchor cell, AC. This will, after further division, it will form an anchor cell, and this will form ventral uterine. So this was the process of formation of the anchor cells. Hope you have liked the video, and if you like the video, please don't forget to share, like and subscribe. Thank you. Thanks for watching.